Hello, my name is Chris Murray, and I'll be talking about one of my favorite animals, the sloth, and why it's so slow. So sloths live in South American rainforests, where it's about 70 degrees Fahrenheit with 90% humidity. They spend about 20 hours of their day sleeping and most of their lives in trees. They only climb down to the forest floor about once a week to urinate and defecate. Sloths are nocturnal animals and are generally solitary except during mating season or if a female is carrying a baby, like the baby in the picture to the side. Sloths have a symbiotic relationship with fungi and algae, which turn their hair green, as seen in the figure below. This helps conceal them from predators such as harpy eagles or jaguars. They also have sloth moths within their fur, which spend their entire life on the sloth. They can contribute to the forest plant life after the sloth climbs down and urinates or defecates on the floor. So there are two distinct species of sloth, each coming from the same order and suborder, however, two different families. The first is the bradypus, which is more commonly known as the three-toed sloth. They are called this because they have 3 claws on the front and hind limb. Their wrists are fused, which allow them to use their hands as grappling hooks to grab branches even better. They have 8 cervical vertebrae, which allows them to rotate their head, and are also blackish in color. This species only eats leaves that are around them in the trees. The colobus, or two-toed sloth, has 2 claws on its front feet and 3 on its back feet. They have unfused wrist bones, which allow them to manipulate food such as fruit, flowers, leaves, or stems. They have 6 cervical vertebrae, which kind of constricts them in their ability to rotate their head and are unable to rotate it to the degree of the three-toed sloth, and are blonde or tan in color. So here are the two families together, so you can notice the 2 different colors and facial structures of each sloth. So there are more breeds per se of sloths under the 2 families I just showed you. There are 2 different two-toed sloths and 4 different three-toed sloths. Each come from a different location, and each have a different category of endangered species assigned to them. For example, the pygmied three-toed sloth is the most critically endangered in this list. So sloths can move at a whopping 3 to 6 hours per mile, so I could probably beat them in the running race, maybe. Some theories as to why they are slow include a poor thermoregulatory system, because sloth's body temperature sort of depends on the environmental temperature. So what I mean by that is if the environment is about 70 degrees, then the sloth is going to be about 3 to 4 degrees higher. If the temperature is about 68 degrees, then it's going to be again 3 to 4 degrees higher, as opposed to our temperature, which stays at a constant rate of 98.7 degrees. They also have a low metabolic rate, where they ingest food, and which will take 1 to 2 weeks to pass through their digestive system and come out as feces. Sloths have a protein-bound iodine, which will cause hypothyroid symptoms and a low overall metabolism. When exposed to stimuli similar to a predator, sloths had little to no response in scientific experiments. It was thought that the sloth's response is an evolutionary mechanism where they chose to ball up instead of fight. And lastly, the sloth has a unique skeletal muscle composition, which I will get into shortly. But before we do, let's review some concepts of muscle contraction that we learned through in class. The sarcomere is the structural unit of contraction and consists of myofibrils. These myofibrils are composed of proteins actin and myosin. Muscle contraction starts when calcium ions are released and then bind to troponin, which then aligns the muscle myosin head with the actin filament. When the myosin head hydrolyzes ATP, it reorients itself and binds to actin to form a cross bridge. After this bridge is formed, the myosin head pulls towards the center of the sarcomere and then binds to ATP again to detach from actin, and this process occurs over and over again. So a recent study looked at muscle composition in sloths and found that they had a lot of type 1 and type 2A fibers within their arms. The type 1 are slow oxidative fibers and are used to enhance forces produced by the type 2A. The type 2A fibers are a faster contraction time and are also known as fast twitch highly oxidative fibers. Both of these fibers are fatigue resistant, which is good for endurance and for helping the sloth hold onto trees. In this next study, the authors measured the response time in sloths and found the action potential speed from the brain to the actual muscle was normal. However, the muscle contraction itself was slow. They believed it was due to the myosin activity and not a delay in the sarcomere's ability to pass on the signal to the next cell. They did quick local dissections and measured the activity of ATPase, which is an enzyme that catalyzes the conversion of ATP to ADP. This conversion is important in muscle contraction and initiates the binding of the myosin head to actin. So just like the first study, these authors discovered that the sloth had more type 1 fibers, especially in the arm and leg muscles. This would make sense because those muscles are needed to hold the sloth in the tree for hours at a time, and type 1 are slow twitch and endurance fibers. The jaw muscle is the only group with more type 2 fibers, and this could possibly be due to the constant use of those muscles. This study also found the activity of ATPase activated by either EDTA or calcium. The activity of ATPase was slower than the activity in the rabbit. I'm comparing the sloth red fibers, sloth to the red fibers of the rabbit because type 1 fibers are also known as red fibers, and these fibers are abundant within the chest and leg of the sloth as seen in the previous slide. Both species have a lower amount of ATPase. Therefore, there is less ATP being converted to ADP and less energy available for the myosin head to attach. The measurement of aldolase, which is a glycolytic enzyme used by white fibers, is also strengthening the data found in the previous slide. There is low aldolase activity within the sloth muscles, which mean they don't really have a lot of white fibers, which means that they must have more red fibers. The aldolase activity within the white fibers of the rabbit shows that aldolase can be present in high quantities within this type of muscle fiber. So this study concluded that the sloth was slow because of its lower ATPase activity of its muscles. They did not detect any myotonic responses, which mean that the sloth did not experience any delays in the relaxation of their muscles. And overall, the ATPase activity of the sloths were far lower than the rabbit red muscle ATPase activities. In the second study, muscles from both cadaver cats and sloths were dissected to find the ATPase activity within. Superprecipitation of the myosin was also determined, which is used to resemble the normal contracting muscle system. This involves a reaction where the final rate is determined, or finishing time is equal to the reaction when it's 50% complete. This study found that sloths had a much slower contraction time when compared to cats. For example, the muscle in the front of the lower leg of a sloth took 106 milliseconds to contract when compared to a cat, where it took 22.5 milliseconds to contract. Their actin activity was also lower, indicating the myosin head was moving slower. So the example, again, of the front lower leg, the sloth had a concentration of 0.12 milligrams per minute of actin, while the cat had 0.5. Calcium was also lower in sloths, which can indicate that they have a slower or lower amount of calcium being released into the muscle cells, which can also slow the rate of contraction. So in our example of the lower leg, the sloth had a 0.09 concentration of calcium, while the cat had a much higher number of 0.24 milligrams per minute. EDTA, which is also a muscle contraction enzyme, was lower in the sloth, which indicates that there is a lower amount of these enzymes present to catalyze the contraction and make it go faster. So in our front lower leg, the sloth had 0.22 milligrams per minute of EDTA, while the cat had 0.5. Therefore, the cat having more EDTA means that the contraction times will be catalyzed and occur faster. So with the slower contraction time, the sloth had a lower superprecipitation time. The cats averaged about 5 seconds, while sloths averaged about 24 seconds. This test is supposed to resemble muscle contraction and indicates that the sloth takes longer for ATP to be utilized. The study also looked at the sarcoplasmic reticulum, which is a specialized smooth ER that regulates calcium ions in the muscle cells. You can see the sarcoplasmic reticulum in the figure as the little blue wavy lines. Oxalate was used to catalyze the activities so the scientists could see any changes at a higher degree. So the concentration of ATPase was lower in the sloth with and without oxalate when compared to the rabbit. So for example, the upper arm had 0.13 of ATPase concentration, while the rabbit had 0.34. This shows that there were less ATP being converted because of the lack of this enzyme of ATPase. The calcium uptake rate was slower in the sloth, which indicates that the activity of the sarcoplasmic reticulum is slower than the cat. So you can see in the upper arm again, without oxalate, the sloth has a uptake rate of 0.09, while the rabbit has a rate of 0.16. So the study concluded that the sloth's ATPase activity was 2 to 4 times slower than the cat. This made the sloth's overall muscle contraction 4 to 6 times slower. They believe the sloth is slower because the hydrolysis of ATP, and proposed the sloth had a difference in the substrate bindings involved. These substrate bindings are thought to lessen the ability of ATP hydrolysis. The final study looked at muscle attachment and used cadaver sloths for anatomical evaluations. They trained sloths to move through a high-speed X-ray to capture their changes while moving and compared it to the anatomical investigations from the cadaver sloths. The figure for the sloth in the X-ray can be seen below. They found a muscular sling which has 3 important muscles. The first is the rhomboides, which connects the scapula to the vertebrae. The next is serratus ventralis, which holds up the chest muscle between the forelimbs. And then the M. pectoralis is the chest muscle involved in holding up the chest inside. The sloth overall has very strong flexor muscles, which allows them to extend their arms. Their forelimbs are also very flexible, which allows them to navigate trees with ease. Within the shoulder, there are also some important muscles. One of them includes the pectoralis superficialis anterior, which allows the sloth to retract its arm with a great force. The pectoralis superficialis posterior, as well as the deltoides, is responsible of carrying the weight of the thorax. Overall, the shoulder is made up of mostly type 1 muscles, which allows for endurance and holding the sloth within the tree. The sloth's lower back was also examined, and it was found that there is a thin sheet of muscle fibers as shown in the diagram below. The top figure shows what the muscles look like from the side, and the bottom figure with the bones shows the view if you look down at the sloth. The longissimus muscle, as seen as in the color green, has a very small area and volume when compared to the other muscles. This is different than any other mammal because the longissimus muscle is generally the largest muscle in mammals. Sloth's general body composition includes about 20% bone, about 25% muscle, 20% hair and skin, and about 25% stomach and intestines. It's important to note that the sloth only has 25% of its body composition being muscle, while most other mammals have 60% of their body consisting of muscles. Scientists are currently unsure of why the sloth genetically has so much less muscle when compared to other mammals. So overall, the sloth has thin sheets of muscle fibers and a low muscle mass. Of these muscles, the sloth has a lot of retractor muscles, and any extensor muscles are smaller and used to help the retractor muscles do their job. The sloth has a 4 to 6 times slower muscle contraction than a cat, as well as a 2 to 6 times lower ATPase activity. Sloths mostly have a type 1 fibers, which are good for endurance. So what does this all mean for the sloth? Well, sloths are really good at holding onto trees and pulling itself towards things. So that means that you could probably beat them at a push-up contest, but they will definitely win a pull-up contest. The muscle fibers allow them to remain in the same position for hours. However, this makes them susceptible to predators because they are too slow or unable to fight back. This could possibly be a reason why sloths are coated in green algae to help for camouflage. Do you have any questions? Feel free to ask me in class.